So I'm happy to stand before you. Happy to be along the team. Uh, Dr. Karin Tanino, abiotic physiologist from University of Saskatchewan. Then Engineer Nair. Um, really happy to know that he's working on low light availability situations. These are new aspects for us altogether. Then I wish to thank Dr. Sendil for giving me this opportunity to attend this lecture and stand before you to say a few words. And I'm also thankful to Dr. Bhuminathan and other scientists of Department of Crop Physiology. And I'm really happy to stand before the students of crop physiology as well as Department of Seed Science and Technology. It's like homecoming. Actually, when we were doing our MSc Agri program, most of the courses we used to attend together, physiologist and then seed technologist. At that time, we were in the same directorate, crop management studies, actually. Our professor and head, then Dr. Karivadaraju was an alumni of this department. So we had to have frequent connections between the two departments. Still, my friends Jasmine, uh, Vichi, Dr. Jaikumar, they were our classmates, and we never felt that we were two different departments. So I'm really happy when I got the invitation from Dr. Sendil and Dr. Bhuminathan to attend this program. I was really, very happy, and thank you so much for giving me this opportunity. Then when I got that letter that someone is speaking about seed treatment, I really got excited. And really, I'm happy that Dr. Carino has chosen this topic of a novel one-time catalytic seed treatment that induces both short-term and long-term responses in wide range of crops. It seems to be a very wide topic as well. And the terminologies are very new to us, catalytic seed treatment. So I'm really eager to know what are the aspects that you will be teaching us during this lecture. Actually, seed technology department is more oriented towards the seed treatments. Seed treatments in the sense we study more about the technologies that are used to increase the seed germination and seedling vigor, basically. And we also try to develop some technologies that can improve the storability of seeds by reducing the seed deterioration process. So when we handle courses, we talk a lot about the soaking treatments, which was developed by Dr. Will in the year 1883, followed by him Henkel was developing the pre-sowing seed hardening methods. He's a Russian scientist. Then followed by him, it was Hedecker who developed the priming, osmotic priming technology during 1973. So all these names are engraved in the minds of the seed science and technologies. But basically, everything is only to improve the seed germination and seedling vigor. So beyond that, there is a doubt whether the seed treatments can improve the crop growth until the productivity. So that one question was there in our mind. But recently we have developed a seed coating technology, which is commercialized in the name of TNAU Vidayamritam. To translate in English, it is seed elixir. So this particular seed coating, film coating methodology and the product, it helps to improve the speed of germination, seed germination, root architecture, because the secondary roots are increased to a larger extent. It increases the stress tolerance with respect to, as you say, abiotic stress tolerance like drought. So now we are taking it to the field, and many of the farmers have given the feedback that it induces drought tolerance as well as it also increases the yield. So in that way, we are happy that a seed treatment itself can be having a significant say on the crop productivity. So otherwise, we will say that seed treatment can increase the field establishment, the number of plants per meter square, and that will have an indirect effect on the crop yield. But because of this treatment, we came to understand that not only the per area unit productivity, but the per plant productivity can also be increased through the seed treatments. Then Besides that, we have developed technologies such as seed pelleting technology, as it is used by many companies like Infotech and so many other European and then companies based in US also. So I can say that we are the only public sector institute which has developed the indigenous seed pelleting technology. So the technology is basically to increase the seed size and weight so that mechanical sowing will be amenable. So a very small seed of like sesamum can be increased in size and weight. The weight will increase by 8 times. So therefore, it is coated with inert materials to increase the improve the size, shape, and then weight, so that mechanical seeding will be very useful, and precision sowing is possible. Thereby, we can reduce the seed rate. So these are the significance of seed pelleting. So the department has developed technologies such as seed coating and pelleting. Besides that, we have worked a lot on seed priming also. For the past 20 years, almost for every crop, we have the protocol of how to prime the seeds. Since priming is actually soaking and drying, it is a cumbersome process, but coating is very, very amenable for even the seed companies. So as an online process that can be implemented, and we can get almost 75% of effect of seed priming. So that is being welcomed by the seed companies as well as the farmers as well. So I'm in that way, I want to know new things from you, Dr. Karin, about how we can improvise our own research on seed treatment technologies. So with these few words, I once again thank the professor and head and Dr. Karin and then Dr. Neuer for giving me this opportunity to share with you some of my random thoughts. Actually, traditionally in India, seed treatment has been followed. Traditionally they used to soak the seeds in milk or even honey, or even coat the seeds with wood ash. So these are the certain things that were randomly followed by all the farmers to improve the seed germination and then the vigor of the seedlings. In that way, I would like to know what new things we can learn. One time when I went to some lecture in a tribal area, the tribal people used to soak the seeds in some material which can even induce the flowering of the trees. Normally, if a tree takes 5 years to flower, this particular seed treatment can induce flowering even the third year of the sowing. So I do not know how a seed treatment can influence to such an extent, but still there are technologies which we may be we may require to explore those things in future. We need to search things from the tribal population, which are still following the ancestral knowledge. So thank you so much for the opportunity. I'm really eager to listen to Dr. Karin.
She's a full-time professor at University of Saskatchewan. She completed her BSc and MSc at Crop Science at University of Gulf. Graduated with her PhD Horticulture Science at Oregon State University in 1990. Since 1988-89, she has been a faculty member of Department of Plant Science, and in 2006 to 2007, she was acting as Associate Dean. Her area of research has focused on plant abiotic stress, and she was one of the first plant scientists to have used Canadian light source synchrotron. She has an active research program in both basic and applied areas, with projects ranging from seed treatment to induce early germination and root growth, the role of apoplastic and epicuticular barriers in plant stress avoidance, and the mechanism of low light resistance, the role of plant stress in developmental process, temperature mediated bud dormancy induction, strawberry epigenetics, as well as the projects addressing northern food security. She holds W.J. White Professorship, was the second person to have been designated a Global Fellow of Iwet University, Japan. And she was the president of Canadian Society for Horticulture Science in 2016 to 2018. And in 2022, she was honored with the award of the 7 most influential women in Canadian agriculture. And it's time to get into the lecture. So I wish to invite the speaker, Dr. Karen, to deliver the.
You can hear me at the back. My voice is a little rough this morning. Thank you so much for your very nice introductions. First of all, I really should thank Professor Bhuminathan, who invited me to come here. And also to Dr. Rakesh, who has been really helping us over the last couple of days. Thank you also to Department Head Senthil. Hopefully I pronounced that correctly, who I just met today. And also to the Director of Seed Science and Technology. I'm very happy to have been the vehicle by which both of your departments have come together again. This is actually my second time that I'm on this campus. My first time was about 15 years ago. And that was when I met the VC, Abramaswamy, and the registrar, Dr. Sandana Krishnan, who were very influential. And I was very impressed with the campus. And on my second trip, I continued to be very impressed. And one of the reasons is this collaboration between the various departments and the application. For example, the elixir that was just mentioned, or the tonics, and I see the list, 7 or 8 different products that your university has produced and really helps the farmers. And that's something that we really do not do enough in Canada. It's more the large corporations and companies that reap all the benefits. And the universities do not commercialize as much. But the farmers do tend to rely and credit more the universities and trust more the universities. So it makes sense that the university should get more involved in this. So as was mentioned, this is a patented catalytic seed treatment. It's non-enzymatic. It's a pure catalytic reaction, and I'll be going through that. But before I do, I thought I would just since you may not know where the University of Saskatchewan is, so this is, as you can tell, very gem. So Coimbatore is someplace in here, and Saskatoon is someplace in here. I learned this morning from Dr. Senthil that you're planning to visit University of Guelph and perhaps sign an MOU. So I decided to put this map up here. This is where the University of Guelph is in Ontario. Toronto is about an hour nearby. And this is where we are in Saskatchewan. This is 49 degrees north latitude. This is 60 degrees north latitude. I understand you're at about 11 degrees north latitude here. So you're way down south. This is the border with the USA. So our nearest neighbor is the USA. We have 40 million population in Canada. 80% of that 40 million live within 100 kilometers of this US border. So that gives you a broad idea of our country, which is the second largest in the world. I live in Saskatchewan. Saskatchewan is known to children as the easiest province or easiest state to draw. Because as you can see, it's pretty, it's almost like a rectangle here. But we have over 43% of the arable land in Canada is located in Saskatchewan. So agriculture is huge. Canada, you may think about this. You may think that we're like this 12 months of the year, but don't fear. It's not 12 months of the year. It's maybe 3 months, maybe 4 months, depends. Oh, okay, thank you. So it's not too bad, but we get this beautiful hoarfrost. This is our total precipitation. This is in rainfall. We get about 1 meter of snow every year. In rainfall, it's about, over the past 5 years, I think it's more accurate not to go 30 years because our climate is changing so rapidly. The last 5 years, about 232 millimeters of rain. You may be wondering about the temperature. Well, these are the extremes. This is the maximum and minimum for the year. And you'll see that this is 50 degrees Celsius, and this is minus 50 degrees Celsius here. But those are the maximum and minimum. Usually it's not that cold, but it gives you an idea. And this is the mean in here. Okay? In fact, we're classified as a short season, semi-arid, temperate continental climate. Right? We grow a lot of wheat. Canola are our main crops. Across the prairies, we have about 20 million acres of wheat and 20 million acres of canola. We have a lot of pulse crops, lentils, chickpeas, peas, so on and so forth. And as I said, we have over 43% of the arable cropland in Canada. I didn't want to show you just the snow pictures. It's quite a beautiful city and a beautiful campus. Our city is about 300,000 in population. We have, even though we're in the middle of Saskatchewan, there's a large South Saskatchewan River. And it really provides a lot of the cultural aspects to the province. I think, we're known as the Paris of the prairies. This is the campus, and it's kind of hidden here, but this is my college. Our campus was established in 1907. Just 1 year after you. I understand your campus was established in 1906. Our province or our state was established in 1905. And our city was established in 1906 and our campus in 1907. So from the very beginning, our university was an important part of the fabric of our province and our city. And agriculture was the first college or faculty on our campus. It was agriculture and arts and science. So from the very beginning, there were 2 foundational colleges, and we were one of them. What drew me to our university was that we're not just the University of Saskatchewan, but we also have the National Research Council, Agriculture and Agri-Food Canada, which Professor Bhuminathan went to and visited as well, and Innovation Place, which is a research park. And here is the Canadian Light Source or synchrotron. It's the only one in Canada, and it was the largest science project in Canada for its time. It allows us to really see more deeply and with greater resolution. So we use it quite a lot in our research studies. So back to the seed treatment. I'd like to give a bit of the history and acknowledge some of the co-patent holders. So usually animal scientists and plant scientists don't get together. But Dr. Olkowski, and his name is hidden here, but Professor Bernard Larveld came to me. They're from the Department of Animal and Poultry Science. They had this novel treatment to break down manure. And they wanted to test to make sure that it was not toxic to seeds, because after all, the intent was to put it on the fields to help to break down the manure more quickly. So they used this treatment on seeds, and instead of being toxic, it in fact enhanced the germination. And they came to me and asked, is this of any interest? And I said, yes, it is. And this treatment is quite cheap. It's simple. It's easy to apply. And it's just a one-time seed application. It can be, we first used it through soaking or priming. But to get it to the field in a practical way, it really needs to be sprayed, especially on our annual crops. Soaking is okay for paddy field rice and that kind of those kinds of crops, but otherwise we need to spray. So the treatment is green. It's compatible for organic agriculture. And as I said, there's a priming effect, and it also induces greater germination on aged seeds. So that can be an advantage. The benefits of germination enhancement, I probably don't need to tell you. In our part of the country, spring is still fairly cold. The snow is just melting. The soil is cold. So we're looking for treatments that will really advance spring seedling emergence and vigor. And the earlier the seedlings can germinate and emerge, the faster they can mature and avoid the midsummer drought and heat stresses. So early establishment in cool soil temperatures is important, getting off to earlier maturity and therefore also harvest before the fall frost and rain. We've tested it on over 30 crops and cultivars. These are some of them here. And the tests were in controlled environment, in greenhouse, phytotron. This is a catalytic reaction. So it's basically hydrogen peroxide and transition metals, iron, copper, etc. And as I said, it's non-enzymatic. It's a pure catalytic reaction. So the seeds, depending upon what type of seeds, they're soaked at room temperature for 1 to 4 hours, or they're sprayed, higher concentration if sprayed. And we, established germination tests in the dark. One thing that we found is that, yes, once this catalytic reaction is launched, there's a huge spike in dissolved oxygen, which lasts for not only many hours and days, but actually weeks. And the treatment induces bubbling in these around these soybean seeds, and this is the control here. It's a Fenton reaction, classic Fenton reaction, and Haber-Weiss. So we think that we're creating almost a perpetual reaction between the two. And the net result is these reactive oxygen species, ROS. And these are hydroxyl radicals, which are very short-lived, but are very potent. So what are some of the effects? I'm only showing you a few. So soybean germination at 20, 15, 10 degrees Celsius, controls in blue, and the treatment is in orange. And the treatment enhances at various temperatures the germination. Canola, even at, this is 4 degrees C, treatment significantly enhances germination, in various cultivars. We looked at growing the seedlings, and this is in soilless mix, and it enhances lateral root growth. This is at 15 degrees Celsius. And then when we grow it at 10 degrees Celsius, it again enhances lateral root growth, and you can see that shoot growth is also enhanced. These results under controlled environment are very consistent. Here's lentil, 15 degrees C, here's control, here's treatment. 10 degrees Celsius. We're always looking for lower temperatures here in our place because of our geography, but yeah, we haven't tested for high temperatures, but that's another area for collaboration. But again, more lateral root growth. Seed germination, here's some more statistics. Here's control. Here's under priming, different temperatures. The lateral root growth and subsequently root dry weight was significantly increased. That's one of the consistent aspects. Root length, sometimes it is, sometimes it's not, but lateral root growth and root dry weight are consistent. Interestingly enough, this catalytic seed treatment induces nodulation in the absence of inoculation. So even without rhizobial inoculation, it induces greater nodulation. So that's something that you may be interested in following up on too. Peas. And this is at 10 degrees Celsius. There weren't sufficient roots to hold on to the soilless mix and control, but here's the treatment, and with greater shoot growth. This is a close-up. The growers were interested in its combination with fungicide, the interaction. Here's control plus fungicide, and here's seed treatment plus fungicide. It also sterilizes the seed surface. I'm always looking to try to reduce the amount of fungal treatment to reduce the heavy metals in the soil and for environmental sustainability. So probably because of the H2O2 component, there is a surface sterilization that occurs. And this is corn seeds. But it's, it's true on all seeds that we've tested, there's that surface sterilization. But the challenge has been to transfer this technology to the field. We see very consistent results in the greenhouse phytotron, in the lab, but trying to transfer it to the field has been more of a challenge. Here's control, the field, and this is Slicer milkfish, which is a forage. And these are the treatment concentration, various concentrations under field conditions. There's some effect, but it's not as consistent as what we see in the growth chamber. In wheat, this is what we see in the growth chamber. Again, very consistent. In the field, we started going to the field. This is Rensong Liu, one of my main technicians who has been doing a lot of this work. We've done work with two cultivars of wheat. 1 hour, just 1 hour of seed soaking. So the title indicated both short and long term. Short term was a germination, long term meaning the lateral root growth. So it always surprised me how this one-time seed treatment can induce such long-term effects on lateral root growth. So in wheat, in the field, there was a stronger effect of planting date and variety or cultivar than the seed treatment. There was no effect on tiller numbers, spikes, flowering, pollination, induced earlier emergence, and greater root and shoot growth at a couple of the concentrations. Here's the field results. We're in heavy clay, so it's very difficult to dig up the roots. Here's control, and this is treatment at different concentrations, 100%, 200%, 300%. There's some effect there in 2017. In 2018, you can see there is some effect. But when we run the statistics, there's a lot of variation, and so we could not pull out significant results. Although visually, we do see there is some effect. Here's another cultivar, Utmost. This is 2017. This is control on the far left, and then 3 concentrations of treatment on the right. And this is 2018, again, control on the far left here, and then 3 concentrations here. So we do see an effect, but there's more variation than under controlled growth chamber conditions. I came to understand the department or Directorate of Seed Science and Technology is very interested in also seed application and formulations. And so I decided last night to stick in some videos on how farmers treat seeds in their field on farm, which might be interesting for you. So here's one. It's kind of noisy, so I should explain first. So these storage containers hold many tons of wheat. They stand about, oh, I don't know, 30 meters high. I mean, it's just, yeah, lots of wheat stored in this. And this is the wheat coming down. So the wheat is coming down and it's carried up through a conveyor belt to a treatment section. There you will see two basically treatment stations, and then with one bin, it looks like a centrifuge. So they're spraying, usually it's their fungicide. This time we also input our seed treatment, but basically it's a spray and the seeds are going around this, it looks like a centrifuge. And then the seeds are carried up through an auger about 10 meters long. And then by the time the seeds get up to the top of that auger, the seeds are dry, and then they fall into the truck. So that's what you'll see. That centrifuge that I was mentioning, the wheat seeds are being put into here, and then the liquid, whether it's fungicide or our seed treatment, is being dosed on them, and then these wheat seeds are being treated. Okay, so once we treated the seed, planted it, and then this is the wheat from those fields. This is control, and on the right is treatment. Yeah? So there is some difference, but it's not as much as we saw in the controlled environment. But there is some, you can see, but there's some variation as well. Here's canola, see here. Results from other labs. It's always nice when other universities can validate our work and other programs. So another professor's master's student was working on aged seeds. And so this catalytic seed treatment enhanced total germination of artificially aged seeds. This is oats, barley, and two different cultivars of each. Control, 0 here. And it's very dose dependent. So at 50% catalytic seed treatment was significant here in this cultivar, but in this cultivar of oat, it was 6%. So that's another aspect that we're trying to work out is, the dosage. Japan. And we published this a few years ago in 2020. And a single seed treatment through hydroxyl radicals increased abiotic stress in Arabidopsis and in rice. Okay. So here's a control. Here's with the Fe as a transition metal. Basically, how do we treat? So the Arabidopsis seeds are then placed into this seed treatment for various lengths of time and incubated and repeated. So here's control, here's, over time. So I'll just skip through some of these because it's fairly similar to what I showed you in the crop section, but this is from Japan. And then time to reach germination is advanced as well. So the mechanism, this brings it back to the Department of Crop Physiology. What is the mechanism of this seed treatment? And it's a catalytic seed treatment that induces reactive oxygen species. And interestingly enough, when you look back, reactive oxygen species have been here from the very beginning. And so it's probably not surprising that it's at the center of mediating the signals of light, wounding, herbicides, senescence, ozone, drought, pathogens, so both biotic as well as abiotic stresses here. And as you know, ROS can be both damaging, but it's also a signaling molecule. So what interested me is that it has potential to really induce resistance to a number of different stresses. Again, going back to the mechanism, there was a paper that was published a few years ago that showed that hydrogen peroxide enhanced ABA catabolism. And as you know, abscisic acid really prolongs the dormancy or reduces germination in seeds, and through nitric oxide. Okay, so nitric oxide, NO, induces ABA catabolism. Okay? And ABA, once you break down ABA, then it's able to be released into germination. So an H2O2 can also induce some GA biosynthesis, which is also related to enhancing germination. So our seed treatment, we've done some preliminary studies, and our seed treatment using nitrate as a proxy for nitric oxide, the treatment initially induces nitric oxide concentrations. Okay? So, and this is the oxygen, dissolved oxygen. As I said, it has a long-term effect on enhancing O2. So the other aspect, this seed treatment induced GA1 and reduced ABA. So ABA catabolism seems to be induced, but thereby reducing ABA and at the same time inducing GA1. So it may be in acting through nitric oxide through enhancing ABA catabolism. We also looked at Arabidopsis mutans, CYP707 series, 707A1, A2, and A3. Excuse me. And these are ABA catabolism mutants. That means these mutants do not break down ABA. But when our seed treatment is added to these mutants, the seeds do germinate. And it depends. Some mutants are more, the treatment is more effective on some mutants than others. But yeah, so that's one aspect as well. Now, what are the gaps? And this is where I think that we can really collaborate and enhance our research together. Optimization in the field, that's something that we still need to do. And, based on my nice tours and information I've been receiving over the last couple of days, I think that you have a very nice program and a system to, test your products on farmers' fields in various locations. And, it has much more potential for success that way. But we also need to figure out why there's a barrier from our lab or greenhouse conditions to the field. So optimizing, is it the application method? Is it the dose? Is it pelleting? Maybe we need to look at pelleting. We've talked about that internally in Canada, but we haven't proceeded with that. What is the interaction with the recommended fungicides? Can we replace fungicides with this catalytic seed treatment? And as I said, there's variation. Should we be selecting locations with greater moisture capacity, or so on and so forth? Storage time. How long can we store this catalytic seed treatment after launching the treatment. We've tested it up to about a week, and after that, it seems to drop down. So again, pelleting may be a better method. The impact on subsequent whole plant stress responses, abiotic as well as biotic, that has not been thoroughly tested. I'm an abiotic stress physiologist, and I would love to look at that aspect as well. So there are a lot of different areas that we can collaborate on. This is part of my group, and this is Rensong Liu, who did a lot of that work. Now, Professor Bhuminathan asked me to talk about the academic programs from my department. I'll just briefly do that because I don't want to cut into my husband's time too much. This is our College of Agriculture and Bioresources on our campus. It's the largest building on our campus. We have 5 departments within our college. You may call them faculties, we call them colleges. But we have 5 departments: animal science, soils, food, economics, and plants. So I'm in the Department of Plant Sciences, and we are the largest department within our college. Here's another picture of part of our building. At the undergraduate level, so we offer Bachelor of Science in Agriculture. We require English, IELTS 6.5 or TOEFL 20 in each category. In India, you have an advantage because often your working language is English. We have agronomy programs, excuse me, applied ecology programs, and you can read faster than I can. Crop science programs, and horticulture science program. Yeah. And that's a program that I'm most associated with. Okay. So we've got 4 majors, main majors. At the MSc and PhD level, in our department, this is just the Department of Plant Sciences, we have 90, We have 90 graduate students in our department, about 90, master's and PhD. We've got all of these programs. Again, the IELTS and TOEFL apply. And excuse me, if accepted, then a stipend is provided, which should cover tuition and base living expenses. I provide my master's students with $27,000 per year Canadian. You need to multiply that by 62 to get the number of rupees, so you can do the math. PhD students have a base of 30,000 Canadian. I do not take away any stipend if they receive scholarships, because otherwise, I feel, what's the point of, applying for scholarships if I'm just going to take away their money? So many of my students, most of my students, actually are making $50,000 Canadian per year, which is significant. Our Department of Plant Sciences is housed in that state-of-the-art agriculture building. We have 180 research labs, 30,000 square foot phytotron. It's the largest conviron facility in the world. We have a 25,000 square foot greenhouse within walking distance. Land base totals about 2,000 hectares of land, some of them right on campus within walking distance. We have the Grains Innovation Quality Lab, which really focuses on value-added quality traits. Crop Development Center, and that's within our Department of Plant Sciences, and it's released over 350, close to 400 varieties right now, or cultivars. We get a lot of royalties from these varieties, and those royalties are shared amongst all groups within our department. And we also have dual degree programs at the PhD level in collaboration with Japan. These are some of my facilities that we just built last year. These are 12 high tunnels. They're automatically controlled for heat stress, drought stress as a rain shelter, and heat by drought stress combination. It's got 3 replications, so 12 high tunnels. And it's the only facility like this in North America or in Australia. So we're the only one so far. And we've grown wheat in here. There are 48 different cultivars or genotypes of wheat. And it's controlled through solar panels. And we've got a little weather station on each, and there are automatic sensors everywhere, and so on and so forth. So anyway, that gives you an idea of one of our facilities. I don't have time to go through them all, but yeah. So that's basically what I thought I would present to you today. And if you have any questions, I'd be happy to entertain any questions, or you can contact me through email as well. Okay, so thanks.
Since you said that it is a hastening process, so will it affect on the shelf life of the seeds, or what will be the probable time at which the seed should be treated, and what for how much time can they be stored?
So you're interested in that aspect too? Different seeds, different crop species were able to be stored for different lengths of time before efficacy was lost. But we haven't had time to really thoroughly check at all the different combinations. But some of our wheat seeds could be stored for up to, I think, 1 month without too much issue in efficacy. But yeah, we haven't really checked everything between cultivars, between so many different crops, the temperature. We only stored it at room temperature. We did not store it at any other temperature. But I think storage is an important component because let's say the farmers treat the seed, but then all of a sudden it rains. And then they cannot plant, and then they have to hold the seed. So a week for sure is fine for most crops. But after that, yeah, some crops can be stored longer, some not. So it's like that.
I think just kind of thing, but ultimately we need higher yield and farmers point of view it's more price. So in that way, this treatment really see how much the yield is, how they are getting the revenue.
The yield. This seed treatment is aimed to increase seedling vigor and increase lateral root growth so that our crops can mature and avoid. It's all about stress avoidance because our stressful times come in July, late July, August, usually about in July. And that's when our yield can really be decreased. So before the stressful times, if our plants can fulfill maturity and avoid stress, then that's one way that farmers will get more yield in the end. You see? So it's all about stress avoidance.